Now it's time to introduce genetic drift, the fourth of the fundamental evolutionary processes or forces. It's the simplest in many respects, and also for beginners the most mysterious. We'll try to make it simple. Drift is an inevitable but random consequence of sampling. Heron and Freeman introduce it with another one of their mouse life cycle diagrams, and they show it as happening in the formation of zygotes, that is, at the time of mating and union of gametes. It can also happen at any other stage in the life cycle, but it's of good choice on their part to show that it happens from the very get-go at the time of zygote formation. They imagine the population of mice has its usual allele frequencies of p equals 0.6 and q equals 0.4 for A1 and A2. Zygotes form at random, and 10 of them are selected to make the next population. How many copies of the A2 allele did we expect? This was a clicker question last year. Let's just walk through it this time. Well, the frequency of A2 is 40% or 0.4. We made 10 diploid zygotes. That's 20 gametes in all, sperms and eggs. So at a frequency of 4%, how many of 20 gametes did we expect to carry A2? Was the answer 4, 6, 8, or 10? Well, it was 8. That's 0.4 times 20, which is the number of gametes that were used. Then we can ask how many did, in this particular instance of this history, how many did we get? Well, looking at the data in this histogram, there are two A2 homozygotes. They have each 2 copies of A2, so that's 4. And then there were 2 heterozygotes. Each has 1 copy of A2, so that's 2 more for a total of 6. The correct answer was to press 3. And what's the final allele frequency of A2? The illustration here assumes that all of the zygotes turn into juveniles, all of the juveniles turn into adults, and the adults then get ready to make gametes, which will turn into zygotes to start the next generation. Their allele frequencies are 0.7 and 0.03, so the correct answer here was 0.03. That's easy. You can read it off the graph or just count up. 4 plus 2 is 6 over 10 is, over 20 is 0.3, 30% frequency of A2. So once again, drift in this small population has produced a large change in the allele frequencies, but in a random direction and for random reasons. This sampling process occurs whenever a finite number of events takes place, even if there is a well-defined and precisely knowable probability with which the alternatives occur. And that's the case in gamete selection. So in a population with 60% of A1, if we start sampling A1 and A2 gametes to make, say, a large number of zygotes, 250 in this figure from the textbook, then although the average expected frequency of A1 is going to be 0.6, we're on average going to pick an A1 gamete 60% of the time. At first, when the sample is still small, it is very unlikely that we will have sampled exactly 60%. Indeed, on the very first trial, it has to be either 0 or 1. In this particular experiment, illustrated in the figure from the textbook, the first to be sampled was clearly an A2 gamete, because the frequency of A1 was 0. Then another zygote was made involving sampling actually 2 gametes, and still the frequency was low, so mostly at first A2 gametes were being selected. Then the frequency shoots up over the next few zygotes and rises above 0.6 for a while, meaning that briefly, anyway, A1 gametes were overrepresented for a while. Then A2 become overrepresented, and the cumulative frequency drops below 0.6 down to actually around 0.5. Then it comes back up toward 0.6, then down a little bit. But notice as the cumulative amount of sampling increases, the wiggles in the line become smaller and smaller, and the line stays very close, if not exactly on, the expected frequency of 60%. And by the end of the experiment, when 250 zygotes have been made, which is to say 500 sperms and eggs have been sampled from the gene pool with a population frequency of 60% A1, the curves completely right over each other, so we can't see the gray expected line underneath the red observed curve. And qualitatively, it would be like this every time we did the experiment, with different details of where it was up and down early on, and then showing a convergence, but not a perfect convergence to the expected frequency of 0.6. Here, for much smaller samples, 10 zygotes, they pick in their illustration from the textbook. That's a total of 20 gametes. The possibilities are anywhere between 0 and 20 copies of the A1 allele. This histogram shows all of the possible outcomes at their theoretically expected frequencies. And getting an exact frequency of 60% is the most probable single outcome, but it only happens about 18% of the time. Being 1 less than 0.6, 1 more than 0.6, or 2 less, or 2 more, or 3 less, or 3 more, those are all very common outcomes. Getting extreme deviations is extremely unlikely, but still possible with such a small sample size. I made similar graphs many years ago. You can tell that by the primitive look of the lettering here. Shows the same idea for 3 different population sizes, 10, 50, and 250 zygotes, being sampled in all 3 cases at an allele frequency of 0.2, a stronger bias. Only 20% of what I call the big A allele, 80% of the little a allele. And notice the n equals 10 case, which corresponds to theirs, has a histogram of expected outcomes qualitatively exactly like this one, with 50 total zygotes, 100 gene copies being sampled. Now the spread of the distribution is much tighter, around 20, the expected number, which is the most common, but not absolutely common outcome, and so on, with 500 gene copies being selected, that is, chosen randomly, to make 250 zygotes. The distribution's even more tightly concentrated around the expectation of 100 copies of big A, but again, there's a substantial spread, and now it's the case that extreme deviations, say down to 10% or up to, heaven forbid, 50 or 100%, are astronomically unlikely. But still, genetic drift is happening. There is no way to stop it. Why does it happen? It happens because populations don't remember where they've been, gene frequency-wise. They remember nothing. Each generation begins from where the previous generation ended, and there is no force that we can imagine or have any evidence for that tends to push the population back to where it was before, unless selection is involved. But if there's no selection, then there's nothing to make the population tend to return preferentially to where it was before. Therefore, drift is equally likely to increase or decrease the allele frequency, and in fact it is precisely equally likely to either increase or decrease going from the current generation to the next. And that happens over and over again, always starting from where the population is now, then going up or down, equally likely, then starting from there, doing it again over and over. That's the behavior that explains the look of these population trajectories, and that leads to a highly predictable tendency for diversity to be lost on average, and for heterozygosity therefore to decline, homozygosity to increase. So random allele frequency changes build on each other and eventually lead, unless mutation or selection or migration intervenes, the drift will eventually lead to the fixation of one allele or the other among those present in the population at any given time. These figures from your book, this pair, shows trajectories of random drift starting from allele frequencies of 50% A1, 50% A2 in very small populations, 4 diploid zygotes, 8 gene copies in all. It shows 10 such histories. All of them, as it happens, go to fixation for one allele or the other within 19 generations. The one case that lasted the longest is shown here in red. The lower frequency figure, lower half of the figure, shows the average heterozygosity over all 10 of these experiments. That's the heavy blue line, and even though some of them decline in frequency, then increase, and so on average, the ensemble is almost continually declining in heterozygosity and following this theoretical curve, the thin gray line, quite closely. Rates of random allele frequency change, and hence loss of heterozygosity, are inversely proportional to population size. And that's shown in this comparison of figures like the previous one. This is the case n equal, where the population size, big N, is 40 individuals per generation. Again, we're plotting the frequency of A1 against the number of generations for a total of 100 generations, and doing 10 random runs of this history from an initial frequency of 0.5. Now, quite a few of them are still polymorphic, even at 100 generations, but a fair number, at least 4 or 5 of the 10, have fixed earlier on. The blue curve shows the average heterozygosity over all 10 of these, and the solid gray line shows the theoretical expectation again. And here it is an order of magnitude larger again, from 4 to 40 and now to 400. In this case, all 10 of the populations are still polymorphic for the two alleles at 100 generations, although a few of them have drifted to frequencies closer to the edges, closer to the boundaries, that is to fixation, than they are to the original starting frequency of 0.5. The expected rate of decline of heterozygosity is correspondingly lower, and the observed average for this particular set of 10 histories follows it quite nicely. Many years ago, deep in the 20th century, Peter Bury did a wonderful immortal experiment of this sort with actual organisms, fruit flies, Drosophila melanogaster, that were polymorphic for a brown eye color mutant. He started 107 very small populations set up with 8 male and 8 female heterozygotes for the brown and the brown 75 allele, which can be scored just by looking at the eye color of the flies. So it was a very low-tech way of just keeping track generation by generation where each of these 107 isolated populations had got to on the scale of possible allele frequencies, which is shown here on the x-axis, generation after generation. And he heroically did this for a total of 20 generations. Generation 0, which we don't see, that's the one where everybody is a heterozygote, and so the allele frequency distribution is just a spike here in the middle at a frequency of 0.5, and then the next 19, generations 1, 2, 3, 4, and so on. Every generation, he randomly selected 8 males and 8 females produced in the previous generation and established a new vial with them, and then just rinse and repeat for 19 cycles. And it's a fascinating demonstration that this theory of random genetic drift really does apply to the world. So here in the first generation, these rather small populations have spread out in allele frequency quite a bit, but none have yet fixed. Likewise in generation 2, 3, and finally in generation 4, one vial, it appears, fixes for the BW75 allele. Another one does in generation 5, and then maybe another or so in generation 6, a couple more in 8, 9, 10, and so on. And once we're up beyond generation 10, the largest categories are the largest classes are the vials that have fixed for either the BW allele or the BW75 allele. But notice, as expected, on average, it is equally likely that they fix for one or the other. And all the other unfixed populations come to resemble a completely uniform distribution of allele frequencies anywhere between 0 and 1. And that is exactly what theoretical models predict should happen. So the allele frequencies diverge randomly, but the average overall heterozygosity decreased steadily in almost, but not quite, every generation. Especially here in the beginning, there was a down and an up, and then it was pretty much down all the way. This figure plots the decrease expected in a simple mathematical model of this universe for populations of 16 flies, and notice the real decline in heterozygosity was quite obviously a bit faster than that, which is a very interesting problem in biology. You should think about possible explanations for that. I don't remember whether Heron and Freeman discuss it. It turns out that this estimate just got by fitting a theoretical curve to the last few generations, suggests that the effective population size in these vials of 16 flies was actually closer to 9. But in any case, qualitatively, the decline in heterozygosity was just what was expected for a small population of the order of 16 flies. Okay, so applying this to the real world, we immediately realize one prediction would be that little populations will lose variation faster than large ones, so they ought to have less variation at equilibrium under the combined effects of all the so-called forces or processes that we've introduced: mutation, selection, migration, and drift. Again, the textbook has this a nice review of a study of 4 different plants that were heroically surveyed. They all had populations that could be identified over several orders of magnitude of apparent average size. These were habitat patches suitable for these plants. The plants are shown here on the left. These 3 are herbaceous, flowers, we might call them, and one was a eucalyptus tree in Australia, Eucalyptus albans. And the data for all four of them are shown in the next few slides. So the plots all show an estimate of the population size on a logarithmic scale, and then two measures of genetic diversity. Polymorphism means the proportion of loci. This was enzyme polymorphisms, many different loci surveyed at enormous effort. So polymorphism means what fraction of all the loci surveyed were polymorphic detectably in each population. And allelic richness, the measure shown here on the right, is the average number of alleles per locus. So it's got to be a number greater than 1, but it's never actually very much greater than 1, because some loci are fixed and others have 2 or 3 or 4 alleles. And it's true for all 4 species. The relationship is positive and significant in almost every case. It's always positive and significant in most cases for both polymorphism and allelic richness, that as the estimated size is of the local populations get larger, there's more polymorphism, that is a larger fraction of polymorphic loci, and the average number of alleles per locus also goes up. Eucalyptus is the one that's a tree. It's quantitatively different from the others, and it has a number of small populations that were quite close to each other, less than 250 meters from other populations. That's these blue ones, and interestingly, they all fall above the line, whereas the similarly small populations that were more isolated all fall a bit below the line. Also, just what we might expect based on the idea that these populations close to each other will have been exchanging genes more often, and as we saw, that will tend to give them a larger number of alleles at any given time. Eucalyptus, the tree, is different in that it has absolutely more polymorphism, higher fraction of loci that are polymorphic, and a larger allelic richness than the others, right? With allelic richness numbers in the range 2 to 3 as opposed to 1 to 2, and also a much higher fraction of polymorphic loci. What do you think might explain that difference? An interesting question to contemplate that may end up on the quiz. And I think we'll stop there, take a break, and come back to talk about the interaction between selection and drift in the last segment.